All right. Welcome, everybody, to Spinal Cast, hosted by the Morton Care Paralysis Fund. I'm pleased to be able to bring to you today Dr. Sam Wattrus of Harvard and Massachusetts General. Today we're going to talk a little bit about the research that MCPF has supported and also about Sam's thoughts of all the research going on and a little bit about the future. If anyone has any questions, please go ahead and submit them to the chat, and we'll be taking questions at the end. Sam is a junior fellow in the Department of Molecular Biology at Massachusetts General. His research group utilizes a combination of genetics, lineage tracing, and live cell imaging in zebrafish to understand how cells interact during development and regeneration. Sam received his undergraduate, master's, and PhD degrees from Harvard. He was part of Forbes 30 Under 30 in Science in 2024, and he received the inaugural MCPF New Investigator Grant. Sam, welcome to Spinalcast.
Thanks so much. It's really nice to be here with you.
Well, what we'd like to do, as I said, is chat a little bit about you and the work that you do. So if you're good with that, we'll jump right in.
Sounds great.
First, why don't you tell me a little bit about how you got into this field? How did you end up where you are and studying what you study?
Sure. I'm classically trained as a developmental biologist, but I've always kind of wanted to study the whole range of what that means. When I was an undergraduate, I was introduced to sort of how cool it is that embryos know how to build themselves. You go from a single fertilized cell to this complex multicellular organism. And that requires necessarily a lot of different interactions to take place, a lot of information to be processed to build all these different tissues. And that's sort of endlessly fascinating. But I also think that, and a lot of people agree, like this lends itself to asking sort of the next question of, well, if we did it once during early development, could we do it again in the context of regeneration? And there's a ton of different species out there, including humans to some degree, that can regenerate a number of different organ systems. And so it feels like a very natural fit to sort of look out in sort of the wild west of different organisms and see, this salamander can regrow its arm after you cut it off. The zebrafish can regrow its heart when you give it a heart injury. Like, why can they do this? Why can't we? If these are such excellent models for studying early developmental biology, might they also be great tools for studying regeneration? So I come to studying regeneration and spinal cord injury by way of being sort of a student of the embryo and wanting to know, how do you go from one cell to many cells to complicated tissue?
It's really interesting that there are species out there that can regrow the spinal cord and so forth, just because throughout human history, that's something that we've always just thought it's impossible. And then you realize that there are other species that do that quite easily. It's a little bit surprising at first. So tell me, in simple terms, a little bit about the work that MCPF supported.
Sure. So I'm, as the award name implies, right? I'm still an early investigator, an early career group leader. So I started my lab a couple years ago, and in setting up the lab, I knew that I wanted to work on regenerative biology and knew that I wanted to start by looking in spinal cord regeneration. And I come from my background training in working with zebrafish as a developmental system. So in my PhD training, I had looked at the ways that immune cells interact with the developing blood system and saw these really interesting cell-cell interactions that would take place where the immune system would essentially instruct the stem cells of the developing blood system to do particular things and to act in particular ways. And as anybody who's looked in injury and regeneration settings knows, the immune system is like a big part and a big player in understanding injury and regeneration. And so we decided to take a look at any parallel processes that we could see happening in wound healing in zebrafish. So I mentioned it in passing, but zebrafish are wonderfully regenerative animals. They can regenerate a bunch of different organ systems, one of them being the spinal cord. And one of the things that I saw very early was that you have really profound cell-cell interactions that take place between immune cells and the glia and neurons at the site of these spinal cord injuries. And they looked very similar to the types of interactions I had studied in development. So we decided to start to interrogate that a little bit further. I have some images that might be useful. Let me show you. So this, just for context for people who've never looked at a zebrafish embryo before, what I'm showing you here is just a 24-hour time lapse of the first day of zebrafish development. This is why we really like to use these animals for studying early events in life. So this is the process of going from one cell to many cells. Now these animals at this stage are maybe 5-6 hours old. They're going through this complex rearrangement of cells. And what you can see is there's going to be the emergence of sort of a head and tail. So the head is this part, if you can see my cursor, and the tail is sort of wrapping around this yolk. And within one day, they go from one single cell to this fully formed organism that can move around, can navigate, receive input from its environment. They're spinning around now because they're swimming and it's a sped up video. And this happens very, very quickly in a very repeatable fashion. And what you can see and hopefully appreciate about this is these organisms, these animals are see-through. They're really, really easy to take nice images of. And so it's possible for us to just look at cells of interest and follow them directly in time and see what interactions take place and manipulate those things directly. So when we wanted to look at spinal cord injury, we could take our animals that in this case have green neurons with a laser, actually give them a very controlled spinal cord injury, and then image the immune cell infiltrate. And so if you look now, I've got an immune cell called macrophages labeled in pink. You can watch as these immune cells sort of are recruited into that wound site and start to make contact with the damaged axons and soma at the location, and follow them over time as individual contacts take place and individual events where a macrophage may choose to engulf a cell and get rid of it, or what we see very often where a macrophage sort of covers over the surface of a surviving glia, surviving neuron, and provide supportive cues to it that sort of stimulate recovery. So we knew that if you got rid of macrophages completely, you would just fully block recovery. So if you take a normally very regenerative zebrafish and you wipe out its immune system, it can no longer rebuild the spinal cord. So something about them is really important. And what we decided to look at was this gene that had been so important in my prior work called Calreticulin. So what I'm showing you here is an example of what happens when you manipulate Calreticulin. On the top, I'm showing you a normal control, unperturbed zebrafish. In pink, I'm just labeling the glial cells of the brain and spinal cord. And in green, I'm labeling, again, macrophages, this immune cell. And you can see they both received spinal cord injuries, this top animal and the bottom animal. You can see that macrophages are sort of swarming into that site. But what I find so fascinating, and this is one of my favorite movies we've ever taken, is on the top, you have this really dense sort of plug of macrophages that really invade that wounded spinal cord tissue. And on the bottom, what we've learned thanks in large part to the funding supported by the MCPF, though you still have this sort of global injury response, when those macrophages arrive to that wounded spinal cord tissue, they don't do the same things. They don't perform the same behaviors. They sort of attend to the peripheral wounded tissue, like the wounded muscle and the wounded skin around that spinal cord injury. But in the absence of this calreticulin gene, they no longer engage with the wounded neurons and glia at the site. So what we wanted to do was really interrogate the function of this calreticulin gene family in regulating both the immune cells and then the recovery of the zebrafish after we make these injuries. And what we've learned is that calreticulin 1 is essential for bringing macrophages into that wound site, but then downstream of that is really important for orchestrating regeneration afterwards. In the absence of calreticulin, these zebrafish have a very hard time recovering. It seems like what we've done is essentially delayed recovery. Zebrafish are so regenerative that it's actually quite hard to fully stop them from recovering. But the activities that need to take place for successful repair to happen need to be very carefully timed. And so what we see is there's a delay in sort of making more neurons, making more glia at that wound site. There's a delay in bridging that wounded tissue. What you rebuild looks worse. It is smaller, and the animals take much, much longer to actually functionally recover. So if you challenge a wounded zebrafish to swim a certain distance, normally a larval zebrafish does a pretty good job within about 3 or 4 days of injury. It can swim like a normal, like uninjured fish can. But when you manipulate calreticulin in this way, they're still much, much worse than those unaffected control fish.
It's really interesting. Tell me that it was really valuable, I think, Sam, that you showed us what's happening there. It's a lot easier for people to understand what's actually happening. And if you could contrast that and talk about calreticulin and so forth, macrophages as they relate to human spinal cord injury.
Sure. So, I mean, part of why this topic is really interesting to me is zebrafish, for all their incredible, sorry, I keep looking over here because there's a pictures of zebrafish to my left. For all their remarkable sort of regenerative ability, there's so much that they have in common with us that it seems odd that we can't do the same things. It's not like zebrafish possess some special type of macrophage, and it's not that zebrafish have some wholly unique type of neuron or type of glial cell. Rather, it seems that the ways that those cells interact with each other and the ways that they are regulated is just different enough that they can sort of pull off this sort of behavior and we can't. So the macrophages that are responding to injury in the zebrafish seem to come from all over the body. There's certainly, so in spinal cord injury, we often think largely about microglia, which are the resident immune cells of the central nervous system. And zebrafish have microglia, and microglia are a component of that response. But they also show a lot of peripheral macrophages migrating in from the surrounding tissue as well. And we have the same thing that happens in our case. What seems to be coming out of our work and other people's work, one of the big differences is that the regulation of the macrophage biology is different. So after wound healing, characteristically what you see in regenerative systems, like in salamanders or in zebrafish, is there is this very fast, very strong immune response that must be very inflammatory. Like it has to be very inflammatory and sort of in order to handle the immediate damage, the immediate insult. You can imagine your innate immune system is responding and needs to, defend against suddenly invasive pathogens, sort of block off, like, staunch the wound, so to speak. But in regenerative systems, that early robust inflammatory phase sort of quickly transitions to sort of a resolving anti-inflammatory phase. And that's where you start to get sort of these pro-growth, pro-regeneration sort of activities from the immune system. In humans and in non-regenerative species, that switch to a more resolving sort of behavior seems to be goofed up. So we think that it is sort of a prolonged inflammatory phase in mice and in humans that does not sort of permit the switch to a regenerative type behavior. One of the other projects that we have going in my lab right now is to compare the recovery in zebrafish, which, as we just said, are very regenerative, with another species of fish which are much less regenerative, this Japanese rice fish called Medaka. And what we're finding is like that is the primary difference that we see. They have very, very, very similar neuroanatomy, very similar immune profiles, but for whatever reason, the influx of immune cells in the Japanese rice fish is very inflammatory and stays very pro-inflammatory for a long time. And that seems to be the case sort of in these settings of failed, incomplete, scarring recovery.
I see. And it's great that you have other species like that to compare directly against that are so similar. It's got to help. It's got to help a lot. Tell me, if you would. I know the zebrafish has been studied quite a bit over time because of its regenerative properties. Tell me a little bit about how your research is building on that or is different from that.
Mm-hmm. So there's a few things I guess I could say. One of the things, candidly, right, is we've improved the technology in a way that we can do a better job of understanding what's happening. So movies like the ones I just showed you, up until 10, 15 years ago, would have been extremely difficult to acquire. It's much, much, much easier for us to do this kind of time-lapse imaging at scale now. So, we could, in my lab, we have a really powerful microscope that allows us to basically set up and take movies like those ones that I showed you of 40 embryos all at once every day if we wanted to. And capturing sort of the activities of cells live and being able to say what was going on, at time zero, what's going on now that this cell has arrived, and what did it do, is really, really powerful and sort of a thing that I'm always trying to advocate for in biology in general, live imaging. If you think about in the absence of live imaging, the sort of activities of a dynamic cell like a macrophage that comes in, grabs onto another cell and maybe eats it, or maybe, provides growth stimulation to it to make a division happen. You can only see that happen in a time lapse. Whereas if you were doing sort of static sort of sections and staining that some people have to do still, where you're working with sort of like big samples, say like an adult mouse, with those scenarios, you have to basically just like catch frozen moments in time and try and infer what was going on between cell A and cell B. Mm-hmm. But with live imaging, you can kind of happily stumble upon these interesting things. And then sort of going further down the route of like, improved technology, two of the approaches that were really instrumental in sort of putting us on this path were single cell based technologies. So as you can imagine, a zebrafish larvae is quite small. The site of injury that I'm showing you is really, really, really small. There's not that many cells there. And only quite recently has it become possible to basically pull out the cells at the site of something so small like that and profile what's going on at the RNA level, at the protein level, in any meaningful way. So single cell RNA sequencing technology has only been around for, 10, 15 years. And what we did at the start of this project was single cell proteomic technology, which is still sort of an up and coming thing. But that, those kinds of approaches were what allowed us to say, there's variation taking place here. There's, some of the macrophages at this site are doing one behavior, and some of the macrophages at this site are doing a different behavior. We only know that because we were able to look at these profiles of these cells with such sort of fine granularity, and then able to go back in and look in a time lapse sort of fashion and actually observe what happens when you manipulate them. So I would say technology as a whole has been a really important facet to this project and is part of why we're able to actually do this. Otherwise, I think that we've been able to push stuff in this space and with the zebrafish simply because we're able to apply, different ideas to this system that just haven't been used before. So something I haven't brought up yet, but that what we've been trying to do is to tackle the questions about how many unique populations contribute to regeneration in a zebrafish. And to do this, we're making use of this lineage tracing technology that just simply did not exist before as well and had not been applied in these settings. So what we can do is we can use an approach called Brainbow and apply it in this system where essentially we give every single cell of interest a unique color, perturb the system, which in our case means, make an injury, and then follow that over time and see how many unique populations contribute to regeneration. So something like that, just hadn't been tried here before.
Right. They just didn't have the capability technologically to do that kind of a study before. And now we have that capability. It's really quite incredible to think about the explosion of technology, even, as you said, last 5, 10, 15 years. Technology is very, very different than it was back then. What other things are you saying from a technology standpoint that's changing the game in terms of your ability to do research?
Well, I think that it's been quite amazing to see the genomic technology and sequencing technology evolve over the span of my career, which is not that long. Just before I started my PhD, people in the zebrafish space had finally started to just completely give up on an old approach called microarrays and move into RNA sequencing as a space. And then basically while I was a graduate student, the single cell sequencing sort of revolution took place. And now that is continued to accelerate, and people are doing single cell sequencing to look at epigenetic states. People have managed to couple single cell sequencing technology to look at metabolic profiles. One of the big things now that has been getting a lot of attention is sort of applying, it's called spatial sequencing or spatial transcriptomics, where essentially you would take a slice of tissue and in situ on that tissue slice sort of perform RNA sequencing. So you could combine sort of the information about where a cell is in space along with the profile of what that cell is doing at the RNA level. So that continues to drive forward, and I think is going to be really impactful in certain spaces, particularly, to be fair, in settings where you may not have access to samples like we do, where we can just literally watch on a microscope. If you work with, say, a mouse or any sort of larger organism where you are restricted in what you're able to visualize at any one time, it's really powerful to take tissue slices and then get incredibly high fidelity information about what every cell is doing in a given field. So there's a lot that's happening on that side, I would say, sort of pushing our ability to profile what cells are doing, where, when. Otherwise, always pushing microscopy has continued to become insanely powerful.
It's really interesting, and as you know, AI is exploding right now. Do you think in your field that your work will be impacted by AI, not just from a productivity standpoint, but actually in terms of the science?
Yes and no. it's certainly impacted and going to be impacted because the technology is sort of just ubiquitous at this point. There are folks I know who are I know in my space and in my work who are true believers. I would count myself more of a skeptic of sort of what, or at this point, what I'm comfortable with having AI do. What I've found is that it has a couple of very specific use cases where, I'm perfectly happy to interact with ChatGPT or Claude when I'm trying to figure out how to make my script work so I can, analyze my data set. I have not so far been super impressed with its ability when I have given it real biology and asked it to sort of, either interpret or ideate on it. There were cases when I, before I had interacted with it much, where I was sort of struggling with a weird result in the lab. And, I was like, oh, I'll just throw this into ChatGPT and see what it comes back with. And it would come back with text that reads fine, but conceptually was total nonsense. And what I find more troubling than the fact that it was wrong was that if I would turn around and said, no, actually, there's 2 copies of every gene, like, you forgot this, it'd be like, oh, you're completely right.
Right.
Actually, here's this other thing. And it would just sort of like roll on to the next thing. But that doesn't mean that AI cannot ever reach a level of sort of doing what we want it to or what we imagine it to. But I think I'm more of the mind that we will figure out what to use it for and how to use it in a way that is actually like safe, meaningful, like, checkable. And it won't necessarily just do our whole job for us now,
Yeah, it's interesting to contemplate when I listen to some of the experts about that. One of the uncertainties right now still remains, can they innovate? Can they essentially, air quotes, think of something that no human has ever thought of something before? And apparently that's yet to be determined. So it'll be interesting to see. Tell me about your thoughts on other spinal cord injury research avenues taking place around the country and the world. What are you excited about?
Well, I try and keep a sort of broad perspective on regenerative biology overall. One of the things that I've been seeing in spinal cord injury that I think is interesting is trying to dig more into the innate shifts that happen in the surviving tissue after an injury. So there's been some work in bias, it was Zebrafish work, that started to identify sort of this regeneration associated state that is innate to these surviving neurons that seems to be very important for orchestrating recovery. So essentially what some folks did was take zebrafish, wound them, profile the populations, and identify this rare surviving set of cells. And they identified them as sort of a local hub of sort of directing information. And they didn't get terribly deep into mechanism in this paper, but they basically figured out that if you mess with these sort of surviving populations, the fish overall would do much worse in sort of recovering and rebuilding tissue. And what I think was really cool there was that you could find really clear parallels between those surviving cells in zebrafish that were essential and similar populations in mice. And I think also they checked human data sets as well. So there's like a very clear cross-species functionality here. And again, it is a question of like, what remains to be the difference between these, or what is needed in addition to this cell state for further action in the non-regenerative settings. Otherwise, when I go to sessions at conferences, some of the stuff that I hear a lot about have to do with like basically different approaches to grafting. Some people are really interested in sort of transplanting different progenitor populations into spinal cord injuries. And trying to develop sort of these relay circuits where you have sort of communication from above the plane of injury into the grafted tissue and then down to the surviving tissue below. And that's, not the work that like I'm working on at all, but it seems to be doing things in the data that they show. So I think that that's interesting. And if it works, is a very intuitive sort of method that you would like to take advantage of. I think in general, my main area of interest that sort of I return to over and over again is trying to understand sort of what makes the wound environment tick. Like what are the things that are so bad in a human or a mouse? What are the things that are good or at least not inhibitory in a zebrafish? What happens if you take here's an experiment we're doing in the lab. Like what would happen if you took a non-regenerative species population of cells and transplant them into a regenerative setting? Like, is it just that I have to put this cell into the right environment and give it the right information and it can participate? Like, this is not a transplant that would survive, but if I put like a human neuron into a zebrafish, like, could it grow across a wound site? There's folks who do stuff like that. That's, I think, really interesting, where they've taken really aggressive approaches to sort of reprogramming neurons in mice, and they're able to get really, really impressive axon growth. If memory serves, that there were issues with sort of functional integration past sort of growth past the wound site, but like they solved the issue of like, getting axon extension, which is obviously like step, 1 or step 100 of a bunch of problems that have to be solved, which I think is really, really neat. So yeah, I think understanding the shifts that are happening to the cells local to the site, and then the interactions that they have with that environment are where my attention is.
That's interesting stuff because, there's always, there's so many avenues and so many problems with injury in the human spinal cord that, but obviously axon growth is one of the key elements that has to be focused on. And, it's, there's so much exploding right now compared to what there was even 10 years ago. So it's really encouraging environment out there. When you think of the future and you think of the immune system, there's the old saying, to a hammer, everything looks like a nail. So I understand that might play here a little bit, but given the work you do and what you understand about the challenges of the human, fixing the human spinal cord, what do you see as the role of the research you're doing and more generally the immune system in curing spinal cord injury?
Well, it seems sort of undeniable that the immune system, if nothing else, is a problem to be overcome. And this is true sort of across regenerative medicine broadly. So, whether you have an injury phenotype that you're dealing with, say in spinal cord injury, or whether you're talking about different disease states that we're trying to cure, like diabetes, right? The immune system is a problem we must solve. I don't think it's a problem, more as like a pretty powerful tool that we just need to understand how to manipulate better. And so there's no question that we need to understand sort of how to talk to these cells and how these cells talk to our population of interest if we ever want to fully tackle these things. So I turned to, my mind goes to diabetes as maybe like one of the best examples of where this logic flows from. What was it, 2013, the paper that was published on making insulin producing beta cells in a dish comes out, and everyone goes, "Oh, we've cured diabetes because now we can make insulin from cells. We can make the cells that are lost in diabetes." But that's not really true, because we still have to sort of get those cells into people and have them not be completely wiped out by the immune system. And so since that time, they've been solving a million different problems. But one of sort of the big ones that's still been there is, we have to get these to not die. Like, we have to either hide these cells from the immune system in a way that is functional, or we have to figure out why they're getting cleared out and what we can do to stop it. Similar things here, like, you know, the glial scar that forms after spinal cord injury, like a major facet of it is immune cells, including macrophages and microglia. So we definitely need to interrogate sort of what they do and what we want them to do. There's really, really interesting stuff that's been done over at her medical school in Jigong He's lab, where they're looking at the nature of the responsive immune cells in a non-regenerative setting in mice. And, you know, in a adult mouse, in a mouse older than 1 day old, basically, spinal cord injury does not heal. But in I want to say it was like a mouse like on day 0, it might have been, very, very early, like an embryonic mouse, spinal cord injury, you get regeneration. And the regeneration in that setting requires interaction from the same innate immune cells that in adulthood sort of seem to be negative. And so, you know, even in a mammalian setting entirely, it's clear that you have information that's required or activity that's required from the immune cells that has to orchestrate this process. And like, that's a good example of where if you knew sort of how to manipulate the timing or the exact profile of those cells, you could probably completely change the output.
Well, that's a great segue to a question we have from the audience, which is, how do you translate the discoveries that you make in terms of the things that you're working on? How does that get translated into a human model at some point?
There's a lot of steps. I think, you know, there's a number of things that come to mind in this setting. So we can take the specific gene we've been studying, right? So calreticulin. Calreticulin is a really basal gene in evolution. Basically, every animal has some kind of calreticulin gene that they express. And it's changed a little bit over evolution. So humans have 2 copies of this gene. I think one of them is actually just kind of poorly named. We have one functional copy of calreticulin that I think about. And zebrafish have 3 copies. They're super similar to each other, like very, very similar at a sequence level, but they're behaving differently. And, you know, one of the things we're curious about is like, oh, if I, you know, this would require an enormous amount of effort, but like if I were to make a zebrafish that only had the human version of this gene, how would it behave? You could never do this experiment, but if you had a human that had the zebrafish version of this gene, what would happen? So what I think needs to take place is a proper accounting of, at the specific level of this protein family that we're looking at, and these cells that we're looking at from this species, what's happening and why is that different from humans? So we are doing experiments where we're trying to sort of see if I give zebrafish cells interactions with human macrophages or flip them around, what's going to take place? If I make zebrafish cells present human calreticulin, what's going to happen? How much of this is dependent on sort of specific residues in that protein family? And how much of this is other factors in the environment that are sort of co-opting this same biology? I think that, you know, in the most simplistic version of events, calreticulin functions as this sort of single factor that goes to the surface of the cell, is recognized by the macrophage, and that leads to interactions that are necessary. If it's as simple as that, then you could imagine a strategy where you figure out what's different about human calreticulin and zebrafish calreticulin. That's, you know, quite a bit of work. You change the human calreticulin in a way to make it look more like the zebrafish calreticulin, or you change the human macrophages to behave more like zebrafish macrophages, and you see what changes sort of follow downstream of that. I think essentially there's a lot that has to happen before this gets to human application. This is very much on the basic biology discovery side, but it fits with a lot of stuff that's happening from other groups in parallel that I think is really important in informing how all of this fits together. So there was a really nice paper that just came out the other day that was looking at just the timing of a lot of things in spinal cord regeneration in zebrafish. And there are different matrix factors that are laid down after an injury takes place. And as with essentially everything we're learning about spinal cord injury and regeneration, it's not so simple as like, it's good or it's bad. It seems like timing is really essential. So they did this beautiful work where they showed that this type of matrix is deposited over the course of regeneration. And if you get rid of it completely, it seems like you have an early response that like looks promising, but then the fish cannot fully recover and actually needs that matrix deposition later. And so there is sort of a graded response that has to take place there where you get this timed production of this factor that in mammals we think is just universally bad, and it probably isn't. In a similar way, I think sort of understanding the timing and the orchestration of the immune cells is quite subtle and requires a lot of sort of poking and prodding to figure out exactly how you want to manipulate it here. It may end up that all of this basic sort of figuring out of like, you know, exactly how this mechanism works here doesn't ultimately have to manipulate these specific factors to be useful in people. Maybe at the end of the day, the Calreticulin project tells you, oh, what you really want is a fast immune response of this particular profile, and you want it to resolve quickly. Maybe you could get all those things to happen in a human without messing with calreticulin. Like maybe if you are simply changing the timing and regimen of different inflammatory, anti-inflammatory drugs, you could manipulate the speed and profile of these cells to sort of mimic that same output.
Gotcha. Appreciate that insight. Well, one last question, which is for our audience and those who will watch this recorded later. What would you counsel people to do if they wanted to support this kind of research and support the work that you do?
Yeah. How do you know what is real and what is a scam?
Well, that's if I can, those are two different questions. Why don't you take the one from the audience, which is, what is real? How do people know what is real and what is a scam in terms of research or trials?
Yeah. I mean, there's two flavors to this. I mean, in terms of what's real and what's a scam in research, that is hard to say when it's, you know, early stages basic science, because sometimes insane sounding things are actually totally for real, and, you know, with time will bear fruit and make sense. And if it's bunk, then we trust that the process of iteration and replication will reveal that something is not real in the lab. On the level of treatments and trials, it's really tough. And, you know, this has always bothered me in the stem cell biology space, because there's a ton of stuff out there that is total snake oil. And it really preys on people who are looking for help. There are groups and resources that I would direct you to. Basically, any scientific society, particularly the large ones, often have pretty good resources for these types of questions. So with stem cell biology, my mind goes to the International Society for Stem Cell Research. They have a very active, they made a very active webpage and team that sort of helps one, advocate to Congress on stem cell science and like running good science and good trials. But also that is a place where people can like report potentially harmful scam trials, and they will look into it and sort of flag it if it is bad. So some scientific societies can sort of provide that service. If one does not exist, or if you can't find it, then my suggestion would be to sort of do your best homework on sort of looking at who's doing this. If it's coming from a seemingly reputable source, again, we try and look for people who have connections to like large academic centers, usually to large reputable scientific societies. I would say NIH supported research is typically where you want to be paying attention to. There are red flags that come up if it's a clinic that is not operating in the continental US but is marketing to people in the continental US who have the finances to fly out to a sketchy secondary location. Probably not a good idea. And actually also, frankly, a lot of people are happy to sort of give you your 2 cents on things. So like if you reach out to anyone with any kind of science background and say like, does this seem for real? This is sort of using your network sort of thing, but often people can give you some good advice that way too. But my suggestion is always to run it by sort of trusted bodies.
That makes a lot of sense. Well, Dr. Wattrus, I want to thank you for taking the time to talk with us today about the great work that you do. I also want to thank you for doing that work. We need people out there like you who are working on our behalf to eventually try to solve this conundrum of spinal cord injury and paralysis. So thank you
Thank you
so
so
much.
much. I really appreciate it.
It's been a pleasure having you on the show. For those of you who are watching now or later, if you like this program, please give it a like and subscribe. And we will notify you when we have our next researcher on the show. Thank you so much, Sam. Appreciate your help.
Thank you so much.
Bye-bye.